Hello and welcome everyone. We're going to continue our discussion of some extensions into Mendel's rules. We're going to start back up talking about lethal alleles and then pleiotropy and phenocopy. Alright, so let's begin talking about lethal alleles. This is a condition which, as you might imagine from the name of it, causes death. So we've talked about allele pairs before, where we have big A, big I gives us maybe a wild type phenotype. Little a maybe gives it as a recessive phenotype. And then the heterozygote often would give us a wild type phenotype as well if this was a recessive case and we weren't talking about any of the extensions. Well, a lethal allele says that one of these, maybe it's the big A, maybe it's the little a, maybe it's one of them that has to be as the homozygous form, but one of these possible genotypes is lethal. So it's not just a phenotype. Well, it is a phenotype, but it's a phenotype that causes death. So in this example, let's just say the little a is the lethal allele, but only in the homozygous recessive genotype. In this case, we would say that the gene A, big A here, this allele, is essential, because if you don't have at least one copy of it, it causes death. In all the other cases we've talked about before, we would say that those genes have been non-essential. If you mutate the Y gene in peas, and you make the pea seeds green, it doesn't kill it, it just makes it a different phenotype. But now we're talking about a mutation that leads to death. Now, it doesn't mean that death occurs immediately. Sometimes it can. It can be something where it causes a lethality where the individual does not pass through embryonic development. So we should talk about this term called age of onset. It could be an embryo. It could be embryonic lethality, meaning that it occurs before development or during early stages of development. Or it could be something that happens as an adult or sometime after the birth of the child. So for instance, embryonic lethality, we'll talk about some examples of this, but one would be the yellow phenotype in mice. Another example would be the Manx cat, which you might have seen before, the tailless cat. In adults, a classic example would be Huntington's disease, where individuals with this lethal allele, this lethal mutation, will die at a much younger age. They'll start having symptoms in their 40s usually, which then leads to death. It might be something like cystic fibrosis, which anymore isn't as lethal as it once was. At one time, somebody with cystic fibrosis probably didn't survive past their teen years. Luckily now with modern medicine, people with cystic fibrosis can live much longer, even into reproductive ages and beyond. So the age of onset is important when we talk about lethal alleles. We should also talk about a term called conditional lethal allele. So if we continue our example up here, an individual who is little a, at one condition, and we'll give an example, say 37 degrees Celsius, say body temperature, is healthy. Yet the same genotype at a different condition, we'll say 42 degrees Celsius, is lethal. There are many examples of this. When we study model organisms, sometimes we'll generate certain alleles, like say in Drosophila, where the fly doesn't exit embryonic development at a higher temperature, but at a lower temperature they do just fine. We see this in humans as well. Sometimes the diet will affect the way a certain allele is expressed. There's an enzyme in our glycolytic pathway called, I might as well write it down, or abbreviated, glucose 6-phosphate dehydrogenase. And if there's a mutation in that, you're typically okay. But Interestingly, if you eat fava beans, it's lethal. Alright, let's erase this. So how do you detect, or how would you predict lethal alleles? Well, let's talk about an experiment that was first done that described lethal alleles for the first time. So there was a scientist in France, his name was Kuno, you don't really need to remember his name, but he was studying mouse coat color. And he was trying to get a pure breeding strain of a yellow coat color, and he could never get it. So he would take a yellow mouse, and he crossed it with a yellow mouse. And yellow, I just mean the coat color of the mouse. And he always got a 3-to-1 ratio of 3 yellow to 1 gray. Even when he came back in here and took these yellow ones and crossed them together, he would still always get a 3-to-1 ratio. Or I should actually put what he thought was a 3-to-1 ratio. So when he saw this 3-to-1 ratio, he presumed that these were heterozygotes, because that's what you would get. You would predict, or what he would have predicted, was that YY and YY gives you yellow, and little y gives you gray. And indeed, when he got this gray one and he crossed it with itself, he got a pure breeding strain of gray, but never could he get a yellow one. So of course the obvious assumption is that Mendel was completely wrong, but that's not true. It turns out that these yellow are always heterozygotes. So let's do a cross here. So he takes these two yellow mice. We now know they're heterozygotes. At the time he didn't know that. And he crossed them together. And let's draw a Punnett square, because we love Punnett squares. I'm going to put a line under the little y just to make sure you know I'm talking about a lowercase y. So he presumed that this one was gray, and he was right. And he presumed that these three would be yellow. Well, it turns out what happens is that indeed this one is yellow, and this one is yellow, but this one is dead. It doesn't survive embryonic development. So he never counted it in his crosses because he never saw it. And the problem was that Kuno didn't have quite the same passion for numbers that Mendel would have. I suspect Mendel would have caught this and realized this really wasn't a 3-to-1 ratio he was counting. He was really counting a 2-to-1 ratio. It was just close enough for Kuno, and he figured that, I guess he figured, that it should be a 3-to-1, because he didn't even think that this was a possibility. Remember, this was in the early days of genetics. He thought it was close enough. He was wrong. It turns out that when you do a cross with a lethal allele, you're always missing one category. It could be, in other examples, that the heterozygote has the lethality, or the homozygous recessive has the lethality. It depends on the case. But in this case here, it was the homozygous dominant that caused the lethality. So when you're looking at a problem, if you're getting a ratio that is not a predicted ratio, and it appears that there is a category missing, as we see in this case here, one assumption could be lethal alleles. Alright, let's go ahead and erase this. And let's talk about this term called pleiotropy. It's always good to go back and look at a simple example here. Remember, we always said that when we're missing a gene, so let's go back to our favorite Mendel peas, the yellow peas. We said that this gave us yellow. And that in the homozygous recessive, it gave you green. One mutation gave you one phenotype. So let's write that here. One mutation equals one phenotype. Pleiotropy would say that, okay, so pleiotropy says that one mutation gives you many phenotypes. So how is this possible? Well, there are several ways that you could have a single gene mutated that's going to give you multiple effects. The first is, it could be that the gene that's mutated affects many different processes. Your book is a really good example here. Is if you had say a microtubule mutated. Well, microtubules are important in many different things. They're important in moving vesicles within the cell. They're important in cell division. So just those two alone, we're going to end up with defects in division, cell division. Presumably you'd still be able to do math, so I'm talking about cell division here. And intracellular, within the cell, the ability to move vesicles around would be affected. And this could lead to quite different phenotypes. And remember, when we were talking about multicellular organisms, the second is when we're talking about more complex organisms that have multiple tissues and multiple organs, you could have the same gene expressed in multiple organs or tissues. So as an example, you may have a gene that's expressed in muscles. The same gene is also expressed in the brain, maybe also in the kidney. Well, these tissues and organs are very different, so lacking this gene in each of these is going to give you a different phenotype. Being a multicellular organism sometimes creates unique problems. And the other one is what we'll call temporal, the timing of the expression. So you may have the same gene expressed as an embryo, a fetus, child, a developing child, and an adult. At each stage of development, this mutation may have a different phenotype. Because of these three reasons, it's common that a mutation may have multiple phenotypes. Alright, let's next talk about something called phenocopy. A few things we should say about phenocopies. First of all, they are not heritable. They are heavily influenced by the environment. So a good definition of it would be a phenocopy mimics a phenotype that can be caused by a gene mutation. A good example is deafness. We know that deafness is linked to a homozygous recessive mutation. We'll just go little a, little a. We also know that sometimes somebody can have big A, no mutations at all, but still are deaf. And one example of this is a rubella infection during pregnancy. Now, generally, this rubella infection has to happen during the first trimester. But when it does, there's an elevated chance of the child being born deaf. This is one of the reasons why an OBGYN will test to see if a pregnant woman has a natural antibody to rubella or has ever been infected by rubella. Because if they have, then they'll have antibodies to fight off rubella to prevent this rubella infection from causing deafness to the child. So this is an example then of how you can get a phenotype but not have the mutation. So this environmental stimulus, in this case an infection, copies the phenotype that is generally caused by a gene mutation. All right, I think that's a good place to end this podcast. In this podcast, we talked about lethal alleles and pleiotropy and phenocopy. In the next podcast, we're going to talk about environmental factors and sex influence and sex limited traits.